asked to give the quiz. The first question here you can find the restriction endonuclease enzyme that can cut 5' GTT AAC 3'. So many of you have responded, and so many of you have responded this as G echo R1, and only less number of you have responded this as Hind 2. Then nobody has Sal 1. So can anybody tell me actually what is the right answer?
Hind 2.
Hind
Hind
2.
two. Hind two.
Yes, it is Hind 2. Okay, it is Hind 2. It is Hind 2. So you can see this is more highlighted here. This Hind 2, it is the right answer. And next question. You please uh, do this quiz. You click on this link that I have sent in your WhatsApp group and please do the answer. So only one has responded and all other need to also respond. All other need to respond. Please be quick. All need to respond. Only 5 students they have responded. And all other you go through your WhatsApp there, you see the link and you respond there. So here is the type of gene transfer technique in which gold particle is coated with rDNA and fired to target cell. So all the 12 students those who have responded here, they are telling that it is gene gun. Do you agree all?
Yes, sir.
Okay, so you all agree. You all agree that it is gene gun. Okay, so it is a gene gun, that is the technique by which we have to coat the gold particle or the tungsten particle, then we fire it to the target cell. The target cell may be plant cell or it may be your animal cell also. Okay, so thank you all for this quiz. And today we are discussing about biotechnological application in agriculture. So we are talking about biotechnology and its application. And you can find biotechnology can be applied in different fields, maybe agriculture, maybe therapeutics, maybe diagnostics, maybe waste treatment, maybe your food processing, bioremediation, energy productions. So in various such fields, generally we use biotechnology. Okay, we use biotechnology in different such fields.
Sir,
And one such your important field here is the agriculture. Okay, Hampit, please tell. You
sir, what is therapeutics?
therapeutics means generally when we can use the biotechnology for treatment of different kind of diseases, for diagnosis of different kind of diseases. That part generally we call as therapeutics. Okay, so
Okay, sir.
therapeutics it
Thank
is
you, sir.
the use of biotechnology in medicine for treatment and also the diagnostic. Okay, so let us first discuss about biotechnological application in agriculture. So you all know about the major type of agriculture that is being followed by most of the farmers in India or most of the people in the world. They generally follow which type of agriculture? Can anybody tell me which type of agriculture generally most of the people in the world they follow? Yes, Madhusmita, can you tell? It is it is your agrochemical based agriculture. Okay, it is agrochemical based agriculture. And generally, in case of your agrochemical based agriculture, what we do? You all know there is more and more use of chemical pesticides. Okay, there is more and more use of your fertilizers, that chemical fertilizers. So this has increased our food production, and Also, you can find the quality of food also it has been increased. And mostly we are relying on your agrochemicals. We generally use more and more fertilizers. We use more and more pesticide, and we get bumper harvest. But you know, these agrochemicals that we are using, the chemical fertilizers that we are using, though they are increasing our production, do you think the food that we are taking are they safer?
No, sir.
Okay, no, it's not safer. Okay, as we are using different kind of chemicals, the soil of the health of the soil, it is depleting. So the condition of the soil is deteriorating day by day because of more and more use of harmful chemicals. Then another option we are left with, that is your organic farming. And in case of organic farming, generally in place of chemical fertilizer, we use different kind of microorganisms as fertilizer. So can you tell me what we call that? When microorganisms like anabina, like mycorrhiza, okay, so like rhizobium, when these microorganisms can be employed in agricultural field for enriching the soil with more and more nitrogen, what we call this?
Biofertilizer.
Organic farming.
That is biofertilizer. So in organic farming, when we employ different microorganism, maybe cyanobacteria, maybe bacteria, maybe your fungus, they generally enrich the soil with various type of nutrient, and that is organic farming. So there we use also the organic pest. But what is the drawback in case of organic farming, and what is the advantage in case of organic farming? Yes, what is
The
the drawback in
drawback
case
is
of organic farming? Yes,
that the production is very very less and very very expensive also.
okay, expensive, very good, and also less production. And what is the advantage? What is the advantage of this organic farming?
Sir, enrichment of the soil quality.
Okay, that health of the soil, that your health card of the soil, that can be maintained for years. That is not possible in case of agrochemical based agriculture. That health card of the soil, it can be maintained for a long time, right? So the chemicals, whatever food material that we are obtaining, maybe fruits, maybe vegetables, maybe cereals. So whatever food material we are obtaining through this organic farming, they are safer. Okay, they are safer. So there is no chance of that biomagnification that is generally predominant in case of your agrochemical based food products like different vegetables, different foods, different cereals, pulses. They're all having more and more amount of that harmful chemicals, and they are entering our body through the food chain and is causing biomagnification. So here also limitation, there are also limitations. You can find limitations everywhere, whether it is agrochemical based agriculture, whether it is your organic farming. Then what is the best option? What is the best agriculture that we have to adopt so that the health of the soil can be maintained, so that the product that we are obtaining, maybe fruits, maybe vegetables, maybe pulses, maybe cereals, that should be also safer from different type of your chemicals. So can you tell me anybody? We have to follow genetically engineered crop based agriculture. Okay, so we have to follow genetically engineered crop based agriculture. Okay, so we have to follow genetically engineered crop based agriculture. Okay, so generally in case of this agriculture, this is the advanced agriculture. This is the your latest agriculture. In case of this latest agriculture, it ensures not only about the health card of the soil, it also ensures your safer products. Okay, it also ensures your safer products without using chemical. We can control different pests. Okay, so the product that we are obtaining, the fruits, vegetables, pulses, cereals, they are generally free from different kind of harmful chemicals. Okay, they are free from harmful chemical. So different advantages are there about this genetically engineered crop-based agriculture. So whatever organisms, whatever plants that we obtain here, those plants, those animals, generally we call them as the GMO. They are genetically modified organism. Okay, they are genetically modified organism. So in case of this GMO, genetically modified organism, genetically, this is genetically modified organism. So can you tell me where such organisms, they are generally called genetically modified organism? Where such organisms, they are called genetically modified organism? Shruti, can you tell? We produce different plant, we produce different kind of your animals, and they are GMO. They are genetically modified organism. Why they are genetically modified? Tanisha, Tanisha, tell. Sneha, can you tell? Sneha, can you hear me?
Yes, sir. Can you please
Huh.
repeat the question?
My question is where the organisms, they are generally called GMO. Why they are so called? Genetically modified organism. So different organism, for example, rosy cow, your BT cotton, BT brinjal, golden rice. So many such organisms are there. So these organisms are generally called a genetically modified organism. So why they are so called?
Because their
Tell why they
genes
are
are
so called?
modified.
Because their genes are modified. So generally you can find that they are allowed to possess alien gene, that gene of interest, and that gene can be expressed in those organisms for a suitable product that we wish to produce. So that product generally we obtain in that genetically modified organism. So the organism, they can possess and express a foreign gene. So that is why they are genetically modified organism. So when we go for production of genetically modified organism, like the transgenic plant. So in case of the plant, we can call them as a transgenic plant. So in case of such plants, transgenic plant, what are the various advantage of producing such transgenic plants or your GMO? What are the various advantage? So many advantages are there. Okay, many advantages are there. So what are the major advantages of producing such, genetically modified organism? Many advantages you can find here. So the important advantages are we can increase, we can improve the nutritional quality. For example, we can increase the vitamin quality. As you know, in case of normal rice, in case of normal rice, can you tell me which food component you can find more in amount in case of normal rice that we consume? That is generally
Carbohydrates.
rich carbohydrate, that is rich in starch. But There is one rice, there is golden rice. That golden rice, this is rich in vitamin A. So whatever component are not present in the your food material, and those food material are essentially required by people, and there is a deficiency of some important component of the food like vitamins. So such important component of the food that can be compensated by inserting that particular gene. Okay, and that can be responsible for the production of biofortified food that will be enriched with the vitamin A or different other such type of your minerals like iron. Okay, so we can fortify that food with the desired minerals, desired vitamin. Then better nitrogen fixation can occur because of such GM crops. Then GM crops that tolerant to different abiotic stresses like cold, drought, salt, heat condition. Then this GM crop, they also reduce reliance on chemical pesticides. So farmers who were using more and more chemical pesticides now, when they are you know they are farming the GM crop. So in case of GM crop, the dependence to the chemicals will be lower. It will be less. So there will be reduction in the reliance on the chemical pesticide. No need of use of that huge amount of chemical pesticides here. Then reduce post harvest losses. So as yesterday I was talking about the tomatoes. So if you are purchasing tomatoes and keeping at home without using refrigerator, then for how long it will be fresh? Can we tell? The tomatoes that we are purchasing from the market, keeping at storing at home, for how many days they remain fresh without using the refrigerator? Yeah, for how many days they remain fresh? Sives, Shreyans Das, can you tell? This is day-to-day activity. You all must have seen tomatoes at home. Madhusmita,
So about 2 to 3 days.
they generally remain fresh for okay less, very less days. There is maybe 2 to 3 days, and in order to keep them fresh for a long time, we need a refrigerator. But nowadays, tomatoes which are available in the market, the tomatoes which are available that can be stored without using refrigerator for even week time. Okay, it can be stored for your week time without using refrigerator. So generally, many such crops are there. Just after the ripening, they are destroyed. But if that ripening period, it can be just enhanced, then that crop losses that can be protected, that can be said, that your crop loss that can be avoided. So reduce post harvest losses, then increase efficiency of mineral usage by plant. So these type of GM crops, they can utilize more and more nutrients. So whatever nutrients are there available in the soil, efficiently those minerals can be utilized by such GM crops. And production of disease resistant crops. And such crops, they do not require any chemical pesticide. Naturally, they can ward off a different kind of different harmful pest. They can protect themselves against different type of your diseases that is infected by fungus or bacteria or virus. Okay, so disease resistant crop that can be produced by uh, through this biotechnology, through this GM crop. Okay, and micropropagation for biomass energy production. So these are the various advantages of this your GMO, your genetically modified organism, genetically modified your crops or genetically modified food we can obtain from them. And in our day-to-day life, the most genetically that modified food we use, mostly that we use, they are soy, corn, rice, canola, tomatoes, potatoes, cotton seeds, papaya, peas, dairy products. So these are the different such product generally we use more and more. In case of pest resistant plants, how can we produce pest resistant plants? You know about different kind of your insect pests are there. Maybe flies, maybe mosquitoes, maybe beetles, maybe your birdworm, maybe your army worm. So different such type of insects are there. Generally, they damage our crop. But through genetic engineering, through biotechnology, we generally produce pest-resistant plants. Okay, we generally produce pest-resistant plants, and this plant, this GM crop, they generally reduce the use of pesticide. For example, in case of your BT cotton, in case of your BT corn, BT rice, BT tomato, BT potato, BT soybean. So in these crops, generally we introduce one gene, and that gene we get from Bacillus thuringiensis. Okay, we get it from Bacillus thuringiensis. So this is the bacterium. It has that particular gene. So we transfer this gene from this we extract the gene, we isolate the gene from this Bacillus thuringiensis, we make the rDNA, then that rDNA through genetic technique, maybe through gene gun, maybe electroporation, maybe micro injection, we transfer to different your crop plants, and we produce the pest resistant plant. So generally BT toxin gene, in case of your BT toxin gene, this can produce one toxin, and this toxin when will be taken up by your plant, when your this insects, different your plant insects, when they consume such your toxin, then they die. Okay, then those organisms, those insects, they die after consumptions of this. So let us discuss how different such your insect. Okay, different such insect, your lepidopterans, coleopterans, and dipterans. So these are the three major group of insects. They generally damage our crops. Under the lepidopterans, tobacco birdworm, armyworm. Then in case of your coleopterans, your beetles, dipterans, flies, and mosquitoes. So these are the different kind of your insects. They damage our crop. And in order to ward off these insects, generally we go for the gene transfer, that BT gene. We transfer the BT gene to this different kind of crops. And when these insects, they feed on these plants. Okay, when they feed on these plants, generally they are killed. Okay, they are killed. So where they are killed, you know where they are killed? When they will feed on the plants, they feed on the leaves, then these insects, they die. So can you tell me where they die? Yeah, anybody? For example, this is the leaf. So when different such insect, okay, when different such insect, they consume this leaf. Okay, different such your insect, they consume this leaf. So let this is your one insect, this is one caterpillar, and when it consumes this leaf, this die. So this plant is your BT plant. It is let BT cotton. Let in case of your BT cotton, when this plant will be eaten up by the organism, by the caterpillar of the particular insect, then what happens here? This leaf, in this leaf you can find that toxin. So as the cotton, this BT cotton plant, it has that particular gene, and that gene, it is expressed and it can produce that toxin. And this toxin actually not the active toxin, it is the prototoxin. Okay, it is the prototoxin, inactive toxin, prototoxin. And This prototoxin, when it will be consumed by this caterpillar, so that chemical will be going inside. Okay, that chemical that will be going inside, and in the midgut of this caterpillar, in the midgut of the caterpillar, you know what happens to this toxin? Yeah, anybody?
So these toxins will release and thus kill the larva.
It is
Yeah,
activated.
anybody?
It is
Yes,
activated.
it is activated. Yes. So it is activated into your active toxin. Okay, it is activated into your active toxin. It is activated into your active toxin. So you can find here that will be activated into your active toxin, and how this activation occurs. What makes this activation of this prototoxin, inactive toxin into your active toxin?
Alkaline pH.
Alkaline medium.
Yes, very good. So it is because of the alkaline medium at the midgut. Okay, it is because of your alkaline medium, your alkaline pH, that prototoxin or inactive toxin generally converted into your active toxin. And because of this, what happens? This chemical, that active one, it can make this cell, swell. So this cell, there'll be swelling of this cell, and there'll be damage of this cell, and as a result, what will happen? There'll be hole here in case of this epithelial cell layer of the your caterpillar. And what happens? So because of this, the organism dies. Okay, because of this organism dies. So it is a prototoxin that is generally produced in the plant, and when the caterpillar, when the insect that will feed on the leaf, then the prototoxin will be going inside, and in the midgut of this organism, it is activated with the help of alkali pH into your active toxins. And this active toxin that will be acting on the epithelial cells there, there'll be those cells, they swell, they damage, and you can find holes there, and this can cause the organism die. Okay, this can cause the organism die. So you all could get this. So this is your BT cotton. This is your BT cotton, where we generally transfer that BT gene, where we transfer the BT gene. So that BT gene is generally we call as cryogene. Okay, that BT gene is generally called as cryogene. And what is the source of this cryogene? From which organism we get this cryogene? Yes,
Bacillus thuringiensis.
yes, it is your Bacillus thuringiensis. Okay, it is your Bacillus thuringiensis. So it is Bacillus thuringiensis. Okay, so it is your Bacillus thuringiensis. This is the bacteria. So this bacteria has this particular gene, your cryogene. So that cryogene generally we transfer this cryogene into the cotton plant, and we make the transgenic plant. We make the GM crop, that is BT cotton. Okay, that is BT cotton. So in case of this cryogene, generally there are three major categories of these cryogenes. Cry 1AC. Okay, you can find three different type of such gene. Cry 1AC, a small c. Cry 1AC. Then another one is cry 2AB. Cry 2AB. So you all please write this. Cry 2AB. And these two genes, they work against cotton bollworms. They work against cotton bollworm. Okay, so these genes generally they work against your cotton bollworm. So in case of the your ball, cotton ball, they are damaged by the your cotton ball worms, and such cotton ball worms that can be controlled by the introduction of this gene cry1ac and cry2ab. And another gene is also there, that is cry1ab. Okay, that is cry1ab. So you all please write this cry1ab. So in case of this cry1ab, this gene can be introduced in the plant against corn borer. So generally in case of your corn plant, in case of your maize plant, when we are introducing this gene, then the corn borer cannot destroy the stem. Okay, they cannot destroy the stem. So that is cry1ab. So here you can find three cries in cry1ac, cry2ab, cry1ab. So this first two, cry1ac and cry2ab, they generally work against bollworms, and the last gene cry1ab that works against the corn borer. Okay, so this is all about the BT cotton. So in order to make the BT cotton, what we do first? Who can answer? Who can answer? In order to make the BT cotton first, what is the first step? What is the first step for making BT cotton? What is the first step? Srikant, what is the first step? What is the first step? Pritipanna, Rahul, in order to make the BT cotton, that normal step that you know in biotechnology, that principles and processes, you already know how to make your the host plant transgenic. How we can go for that? Susri Savitri. Susri Savitri, please tell. How can you make BT cotton plant? The first thing will be the first step, that you know, the isolation of gene, and gene will be isolated from the Bacillus thuringiensis, and this gene that can be used for making the rDNA. Okay, so we need one vector. So can you tell me which vector we can use here? Anybody? Can you name the vector here?
Agrobacterium
Which vector?
tumefaciens.
Yeah, the vector we obtained from Agrobacterium tumefaciens. And what is that vector? Name of the vector is
TI
TI
plasmid.
plasmid.
TI plasmid.
Okay, so we have to use this TI plasmid, and in the TI plasmid we have to insert this, you know, cryazine. So this cryazine we have to insert here. So cryazine we have to insert here in this TI plasmid, and this TI plasmid. Okay, this TI plasmid for the through different gene transfer techniques, we have to insert it in the host plant. Okay, in the host cell. Getting, so this way we can go for making BT cotton. Okay, we can go for making BT cotton. So this is all about BT cotton, and now you could know how we can control cotton bollworm or different such your worms. This is because this gene, your BT gene, that is the cryazine, it makes prototoxin in the host organism, and when it will be taken off by the pest like the cotton bollworm inside the organism, you can find there'll be your activation of that inactive or protoxin into the active toxin one, and further that can destroy the organism. Okay, so this is the novel strategy to ward off your insect pest like cotton bollworm and many other such insect. Then we have different other your strategy